Welcome to the deep dive. Imagine being able to instantly rewrite the code of, well, actual life. Today, we are plunging headfirst into the world of CRISPR-Cas systems. This is the gene editing tech that's not just rewriting genomes, it feels like it's rewriting the future of medicine itself.
Absolutely. And our sources today, they really give us this stunning overview. We're covering everything, from CRISPR's humble start as a bacterial defense thing,
Right.
all the way to these, frankly, astonishing new versions like prime editing. We'll look at the revolutionary applications in, say, diagnosis and treating human diseases.
And critically, we also have to cover the safety concerns and the ethical challenges. Those have to be addressed globally.
Definitely.
So our mission for you, the listener, is pretty clear. We want to quickly pull out the core knowledge. How does CRISPR actually work? What are the most surprising recent advancements you really need to know about?
And why is it such a game changer for medicine?
Exactly. All while keeping a sharp eye on its current limits and, those tricky ethical lines.
Okay, so to set the stage, let's talk about genome editing itself first. What is it?
Good idea.
At its heart, it's basically the precise modification of genomic DNA right at a specific target spot.
So changing the code.
Yep. We're talking inserting DNA, deleting bits, replacing sections. Usually the goal is either to switch off a faulty gene, maybe add a new trait, or correct a mutation that causes disease.
Now, we have had tools for this before. I remember zinc finger nucleases, ZFNs, and talons.
Oh yeah, the old guard.
But if you look at the source material, it's just crystal clear that CRISPR-Cas systems have completely taken over. And the key insight here isn't just that CRISPR is better, technically, it's that it revolutionized access.
That's exactly right. ZFNs and talons, they were expensive, really difficult to design, often needed very specialized expertise. CRISPR just flipped the script entirely.
How so?
Well, it's got this remarkably simple design. It's low cost, it's highly efficient, and it's quick and repeatable. Suddenly, this elite science became accessible to practically every molecular biology lab on the planet.
That democratization, that's the real power shift. Okay, let's unpack the core mechanism then. It sounds incredibly complex, but the origin story is actually wonderfully pragmatic. Where did this whole genetic scissors idea come from?
What's really fascinating, and you touched on it, is that CRISPR-Cas wasn't, invented in some fancy biotech lab initially. It's an adaptive immune system. It exists naturally in most bacteria and archaea.
It's its day job.
Its day job was protecting these tiny organisms from invaders, mostly viruses, by literally chopping up the viral DNA before it could take over.
So bacteria already figured this out, and scientists basically just hijacked it.
Pretty much. And the core machinery, it's stunningly simple when you break it down. You've got two main bits. First, there's the Cas protein. Think of it as the physical cutting tool, the enzyme. An endonuclease, Cas9 is the famous one.
The scissors.
The scissors, exactly. And second, you have the single guide RNA, or sgRNA.
And the sgRNA is like the GPS system, isn't it? Guiding the scissors.
Perfectly put. The sgRNA is designed, it's programmed, to quickly find and pair up with a very specific sequence on the DNA you want to target. It literally guides Cas9 to the precise spot for the cut.
Okay.
Once it matches, Cas9 makes what's called a double-strand break, a DSB. Gets at both strands of the DNA helix right there.
And that break is the key, right? Because the cell immediately notices and tries to fix it.
Yeah.
And that's our chance to make the change we want. What are the cell's main repair options?
So the cell basically has two main ways to deal with that DSB. The first one, and usually the most common, most efficient method, is called non-homologous end joining, NHEJ. It's active most of the time.
NHEJ sounds like the cell's emergency duct tape. Quick fix.
That's a perfect analogy. It's fast, but it's messy. It's error-prone. It just tries to stick the broken ends back together, often adding or deleting a few random DNA letters. We call those indels.
Indels, okay.
And these indels usually mess up the gene's reading frame, causing a frame shift mutation, which basically inactivates the gene. That's how you get a gene knockout.
Okay, wait. If NHEJ is error-prone, messy duct tape, but it's the cell's go-to repair system, doesn't that fundamentally limit how precise standard Cas9 can be? We're kind of relying on a random outcome most of the time.
That is the exact trade-off, yes.
Yeah.
For real precision, you need the second method, homology-directed repair, or HDR.
HDR.
This pathway is slower, much less efficient than NHEJ, but it's accurate. For HDR to work, we have to provide the cell with a separate piece of DNA, a homologous donor template.
Like a blueprint for the repair.
Exactly. The cell uses that template as a guide to make precise changes, like correcting a typo, or inserting a specific sequence. So NHED for knockouts, HDR for precise fixes, but HDR is harder to achieve.
And we should also mention the gatekeeper, the thing that really restricted early Cas9 use, the PAM sequence.
Oh, definitely. We have to mention PAM. For the most common Cas9, the one from Strep pyogenes, SP-Cas9, it can't just bind anywhere. It needs a specific short sequence called the protospacer adjacent motif, or PAM, right next to the target DNA site.
And what does that sequence look like?
Typically, it's NG. So 2G nucleotides following any other nucleotide. If that NG isn't there, right next to your target. SP-Cas9 generally won't bind, won't cut. This PAM requirement severely limited where in the genome you could actually edit.
So this amazing bacterial system comes with a built-in limitation, that NG gatekeeper, and it mostly relies on a messy repair job. Which brings us nicely to section 2. How did researchers hack past these issues? How did they build a whole toolbox? This is where the innovation really took off.
Right. The innovation was absolutely critical because, the original Cas9 system, it was kind of a sledgehammer. Powerful, but crude. Researchers needed more finesse, like scalpels. And they desperately needed ways around relying on that double-strand break all the time.
Okay, let's start with maybe the simplest tweak. Cas9, dead Cas9. The sources say it has 2 small mutations that basically kill its cutting ability. It can still bind to DNA, but it can't cut. Why would you want scissors that can't cut?
It sounds counterintuitive, right? But you keep the most valuable part, that incredibly precise GPS system, the guide RNA directing it. By taking away the cutting, but keeping the targeting, you turn Cas9 from, let's say, a weapon into more of a programmable delivery vehicle.
Okay, so it's a delivery truck, not a missile anymore. What kind of cargo does this truck deliver?
It can deliver regulatory molecules. So if you fuse Cas9 to proteins that activate genes, transcriptional activators, you get something called CRISPRa.
Oh.
A for activation.
Turning genes on.
Exactly.
Okay.
Or you fuse it to repressors,
Right.
proteins that shut genes down, and you get CRISPRa, I for inhibition.
Turning genes off.
Right. And this lets you control gene transcription levels. You can use it for imaging where genes are located, even for making epigenetic modifications, like adding or removing chemical tags on the DNA. And the key is you're doing all this without permanently changing the underlying DNA sequence.
That's a huge conceptual leap. Control without a permanent cut.
Okay, now let's move to base editing. This tackles the precision problem head-on, the messiness of NHEJ. Base editing systems, things like cytosine base editors, CBEs, and adenine base editors, ABEs, they do something remarkable. They perform highly precise single-letter substitutions in the DNA code. Specifically, C gets changed to a T, or an A gets changed to a G.
And the key mechanism here.
The key is they do this without creating that dangerous double-strand break.
That seems massive, because you completely sidestep the whole messy NHEJ repair pathway.
Exactly. You avoid the cell's unreliable emergency duct tape. So the results are far more predictable, much more precise. The sources suggest base editing, in theory at least, could repair something like 3,956 different gene variants listed in the ClinVar database, which tracks disease mutations. It's like a precise chemical surgery on a single DNA letter.
That is huge potential, but base editing is still limited, right? Just those C to T or A to G changes. Which brings us to what our sources call the ultimate editor maybe, prime editing.
Prime editing is, arguably the most significant leap forward since Cas9 itself first burst onto the scene. It's a bit more complex. It uses a Cas9 enzyme that's been deliberately nicked. It only cuts one DNA strand, not both.
Okay, so no double strand break.
Right. And this nickase Cas9 is fused directly to another enzyme called a reverse transcriptase, and it's guided by a specialized RNA, the prime editing guide RNA, or pegRNA.
PegRNA. And what's special about it?
The pegRNA does 2 jobs. It specifies the target site, like a normal guide RNA, but crucially, it also carries the template for the desired edit directly within its own sequence.
Whoa. Okay, so the guide RNA isn't just the GPS, it's also carrying the instructions for the fix.
Exactly. The reverse transcriptase then uses that RNA template to directly synthesize the corrected DNA sequence right at the target site. This system can mediate targeted insertions, small deletions, and critically all 12 possible types of base-to-base substitutions.
All 12, not just C to T and A to G.
All 12.
Yes.
And like base editing, it does this without needing a double-strand break or a separate donor DNA template.
That just sounds like a fundamental shift in what's possible. If it can do all 12 substitutions plus insertions and deletions and bypasses NHEJ, I mean, where's the catch? It sounds almost too good.
Well, efficiency is still a major area of development and optimization. It's not always as efficient as standard CRISPR or even base editing, yet in all cell types or for all edits. But the theoretical capability is just stunning. One source states prime editing could potentially correct up to 89% of known disease-causing genetic mutations. It gives researchers this incredibly versatile toolkit for precise, predictable editing.
Amazing. Okay, before we move on from innovations, we have to quickly touch on RNA editing. This is different because it's temporary, right?
That's right. This involves the type C CRISPR systems, which use enzymes like Cas13. The key difference is Cas13 targets single-stranded RNA, not the permanent DNA blueprint in the nucleus.
And why edit RNA instead of DNA?
The big advantage is reversibility. RNA molecules are constantly being made and degraded by the cell, so any edits made to RNA are temporary. This avoids the long-term potential risks and, frankly, many of the ethical concerns tied to making permanent changes to the genome.
Makes sense. What's Cas13 used for then?
It's used pretty widely now for things like RNA knockdown, reducing the amount of a specific protein, or for labeling RNA transcripts to see where they are in the cell, and even, as we'll see, for rapid diagnostic tests for viruses.
Okay, so if we connect all these incredible tools to the bigger picture, they're fundamentally changing how we study and even think about treating disease. Let's dig into the applications, starting with disease modeling.
Yeah, genome editing, especially CRISPR, has dramatically sped up how quickly we can create genetically modified animal models. And these models are absolutely essential for understanding how human diseases actually work, before you can even think about clinical trials.
Traditionally, it was mostly mice, right? But the sources seem to emphasize a shift towards using larger animals more now. Why is that?
It's about relevance. Larger animals, particularly pigs and non-human primates like monkeys, are just physiologically and anatomically much closer to humans than mice are. For instance, you mentioned monkeys. Their genomes have about 98% homology with ours. So disease models in these animals often give us much more relevant data for human conditions.
Do we have specific examples where CRISPR has been used to make these more sophisticated models?
Oh, absolutely. Take rabbits, for example. One study mentioned used CRISPR-Cas9 to knock out the PAX4 gene. This gene is involved in pancreatic development.
And the result?
The resulting rabbits developed persistent hyperglycemia, high blood sugar, and other features very typical of human diabetes. It created a valuable new model. And then at the cellular level, another really striking example involves simulating colorectal cancer in a dish.
How on earth do they do that?
It's quite amazing. Researchers took human intestinal stem cells, these are like the master cells of the gut lining, and used CRISPR-Cas9 to edit 4 genes commonly mutated in colorectal cancer: APC, P53, KRAS, and SMA4. And these edited stem cells, grown in culture, spontaneously started developing the biological characteristics of intestinal tumors. It provides this incredible platform to study how cancer starts and progresses, and importantly, to test potential new drugs.
That's powerful. Okay, let's pivot to diagnosis. This is an application that actually uses a really unexpected side effect of some CRISPR enzymes.
Yeah, it's this behavior called collateral cleavage, or sometimes transcleavage.
Yeah.
It turns out that when certain CRISPR enzymes like Cas12 and especially Cas13 find and bind to their specific RNA or DNA target,
They get excited.
they get hyperactive. They start indiscriminately chopping up any nearby RNA molecules, not just the target. Researchers cleverly harness this. They add reporter RNA molecules that release a signal, like fluorescence, only when they're cut.
So the CRISPR finds its target, goes wild cutting nearby reporters, and boom, signal release. It's like a proximity alert.
Precisely. And this became the basis for new diagnostic platforms like Sherlock, which uses Cas13, and Detector, using Cas12.
And Sherlock can even do multiple tests at once.
Yeah, Sherlock version 2, Sherlock AV2, was developed to detect up to 4 different viruses simultaneously in one sample. The huge advantage here is that these CRISPR-based diagnostics are generally inexpensive, they're simple to run, fast, and don't need complex lab equipment.
Which makes them great for
field use, for rapid detection in remote areas or places with limited resources. It's a significant leap beyond traditional, slower, lab-based molecular tests.
Okay, now for the really big one. Gene therapy. Actually treating diseases by editing genes. The strategies detailed in our sources seem to focus heavily on somatic cells. Can you clarify that?
Right. Somatic cell gene therapy means editing genes in the patient's body cells, like blood cells or liver cells. Crucially, these changes are not inherited. They affect only the individual being treated, not their future children. This avoids many of the ethical issues around germline editing.
Got it. So what are some examples in somatic therapy? Let's start with inherited diseases.
For monogenic diseases, meaning caused by a single gene defect like beta thalassemia or sickle cell disease, these affect hemoglobin, the protein carrying oxygen in red blood cells. One really promising strategy uses CRISPR to knock out a gene called BCL11A.
And what is BCL11A?
BCL11A normally acts like a switch, turning off the production of fetal hemoglobin shortly after birth. Fetal hemoglobin, HbF, is the type babies have in the womb and works perfectly fine. By knocking out BCL11A in a patient's blood stem cells, you essentially flip that switch back on.
So you reactivate the body's own backup system.
Exactly. You get the body to produce fetal hemoglobin again, which can compensate for the defective adult hemoglobin, potentially offering a functional cure. This approach is already showing incredible results in clinical trials.
Wow. Okay, what about fighting infectious diseases? HIV was mentioned.
Yes, for HIV, there are a couple of main CRISPR strategies being explored. One is quite direct. Use CRISPR to target the DNA sequences of the HIV virus itself, specifically regions called the long terminal repeats, or LTRs. The goal is to literally chop up and destroy the provirus that's integrated into the host cell's genome.
Eradicating the hidden virus.
That's the idea. The other approach is more preventative, focused on resistance. HIV needs a co-receptor called CCR5 to get into T cells. So another strategy is to use CRISPR to knock out the CCR5 gene in a patient's T cells, essentially locking the door so HIV can't get in.
Making the cells resistant makes sense. And finally, cancer immunotherapy. We're hearing a lot about CAR-T cells, but making them universal sounds like a huge step.
It really is. CAR-T therapy, where you engineer a patient's own T cells to attack their cancer, can be incredibly effective, especially for blood cancers. But right now, it's very personalized, complex, and expensive. Each treatment has to be made individually.
So the goal is off-the-shelf CAR-T.
Exactly. And CRISPR is key to this. The idea is to take T cells from a healthy donor and use CRISPR-Cas9 to make several edits simultaneously. Specifically, they knock out two key genes. First, the gene for the T cell's own endogenous receptor.
Why remove that?
To prevent those donor T cells from attacking the patient's healthy tissues, which is a dangerous complication called graft versus host disease, or GVHD. Second, they knock out genes encoding HLA class I molecules.
And the HLA molecules.
Those are the molecules that flag cells as self. Removing them helps the universal CAR-T cells evade rejection by the patient's immune system. By making these edits, you create a potential universal CAR-T cell product that could, in theory, be manufactured at scale and given to multiple patients. This could dramatically increase accessibility and lower the cost.
That's truly game-changing potential. Okay. Incredible progress. But every story about immense power needs to be balanced. We have to talk about the risks, the limitations, what are the major roadblocks, the safety concerns we absolutely need to discuss.
Yeah, we definitely need to acknowledge that this amazing precision still comes with potential downsides, unintended consequences. The number one safety concern that always comes up is off-target effects.
Meaning the CRISPR cuts somewhere it shouldn't.
Exactly. The guide RNA is designed to be specific, but sometimes it can bind to DNA sequences elsewhere in the genome that are similar, but not identical to the intended target. If Cas9 cuts at these off-target sites, it can cause unintended mutations, potentially leading to problems like cancer or other issues, genomic instability.
That sounds serious. How are scientists trying to minimize that risk? Are we getting better at aiming?
We are, significantly. There are two main strategies. First, scientists have engineered better Cas9 proteins, so-called high-fidelity variants, names like SpCas9 HF1 or ESPCas9. These are designed to be much pickier, less tolerant of mismatches between the guide RNA and the DNA, so they cut only at the intended target much more reliably.
Better scissors.
Better, safer scissors. Second strategy is about delivery. How do you get the CRISPR components into the cells? Delivering them as pre-assembled ribonuclear protein complexes, the Cas9 protein already bound to its guide RNA, called Cas9 RNPs.
RNPs. Okay.
That's highly favored now over using, say, DNA plasmids that instruct the cell to make the CRISPR parts. Why? Because the RNP complex is active immediately, but only lasts for a short time in the cell before it's degraded. This shorter active window drastically reduces the time available for potential off-target cutting to happen.
Okay, so minimizing accidental cuts is key. But beyond cutting in the wrong place, there's also the issue of our own immune system fighting back, right? Immunogenicity.
That's a huge one, yes. Remember, these Cas enzymes, like Cas9, they come from bacteria. They're foreign proteins to our bodies. And it turns out many healthy people already have pre-existing antibodies and immune T cells against common Cas proteins like SpCas9, probably from past bacterial infections.
So our bodies are primed to attack the therapy.
Potentially, yes. This immune response could neutralize the Cas9 enzyme before it even has a chance to edit the target gene, or it could cause harmful inflammation. Overcoming this immunogenicity is a major hurdle for using CRISPR therapeutically in many patients.
And then there's the elephant in the room regarding safety at the target site, the potential cancer risk linked to the cutting itself. This sounds deeply concerning.
It is very serious, and it links directly back to that double-strand break, the DSB, that standard CRISPR-Cas9 makes. Several independent research groups discovered something worrying. These CRISPR-induced DSBs can inadvertently trigger a cellular alarm system called the p53 signaling pathway.
Okay, for listeners maybe not familiar, P53, that's important, right? Why is activating it bad here?
p53 is often called the guardian of the genome. It's a critical tumor suppressor gene. Its job is to detect severe DNA damage, like a DSB. When activated, p53 usually does one of two things. It either pauses the cell cycle to allow time for repair, or, if the damage is too severe, it triggers programmed cell death apoptosis, basically cell suicide.
So the cell kills itself if CRISPR damages it too much. How is that a cancer risk?
Here's the insidious part. If a cell has a working p53 pathway, it often recognizes the CRISPR cut as severe damage and eliminates itself. The danger arises because the cells that survive the CRISPR editing process might be the ones where the p53 pathway is already defective or somehow suppressed. So the CRISPR process itself might inadvertently select for or enrich cells that have lost this crucial safety mechanism. And cells with faulty p53 are known to be much more susceptible to accumulating further mutations and eventually becoming cancerous down the line. It's a potential selection pressure favoring precancerous cells.
That dramatically shifts the risk-benefit calculation, doesn't it? Wow. Okay, we also absolutely cannot discuss CRISPR without hitting the hottest controversy of all, germline editing.
Right. Germline editing means making changes to the genes in human sperm, eggs, or embryos. The absolutely critical point here is that any changes made to the germline are heritable. They would be passed down to all future generations of that individual.
And because the consequences are permanent and potentially unknown.
Exactly. Due to massive safety concerns about unintended, irreversible consequences rippling through generations and profound ethical objections, germline gene editing in humans is strictly banned or heavily regulated in most countries around the world. There's a broad scientific consensus against it currently.
But that scientific red line was crossed in 2018, causing global uproar.
It really did. The controversy exploded in November 2018 when a scientist in China announced the birth of the world's first gene-edited babies, twin girls nicknamed Lulu and Nana. He claimed he had used CRISPR to modify the CCR5 gene in their embryos, supposedly to make them resistant to HIV infection.
And the reaction?
It wasn't just a science headline, it was an ethical bombshell. It ignited immediate, widespread condemnation from scientists, ethicists, and policymakers worldwide. It forced everyone to confront the reality that this technology could be used to make heritable changes, potentially recklessly, and it triggered a massive, urgent global push for stricter regulations, oversight, and a serious discussion about where humanity draws the line.
So wrapping this up, it's this incredible duality, isn't it? This tool offers truly revolutionary potential across research, diagnosis, therapy. We're seeing these amazing innovations like base and prime editing tackling the precision issues.
Absolutely. We're living in a moment of just unprecedented power to control biology at its most fundamental level.
Yeah.
But that incredible power is balanced by really significant safety challenges we've discussed, off-target cuts, immunogenicity, delivery hurdles, that potential p53 activation issue.
Not to mention the profound ethical walls, especially around germline editing, that the scientific community and really all of society must navigate with extreme care and transparency. Which leaves us with a final provocative thought for you, the listener, to consider. We have these rapidly evolving tools, think prime editing, theoretically fixing maybe 89% of known mutations. And we know about these significant risks, immunogenicity, the p53 connection. So how can the global scientific community effectively evaluate the long-term safety and effectiveness of these powerful permanent gene-altering technologies before they get pushed towards widespread clinical use, especially when the stakes involve permanent changes to the human genome?
That's the critical question moving forward. Thank you for joining us on this deep dive.
We'll see you next time.